first speaker today is Brid. She's an investigator in the Laboratory of Human Carcinogenesis. She got her Ph.D. in Ireland, and then she was in the NCI Cancer Prevention Fellowship Program, and then she started to work with Kurt Harris, and now she's an Earl Statman tenure-track investigator at NCI. She's going to talk about cancer health disparities. Brid.
Great. Thanks very much. It's great to be able to give this lecture again this year. I just want to start saying two quick things. So if I speak too fast or if you lose something of what I say, just raise your hand. I'm happy to repeat if necessary or slow down. And also feel free to ask questions as you go through if you want as well. So this is a very detailed topic. It's kind of a complex topic too. So the goal today isn't to cover every aspect of cancer health disparities, but rather the goal is to give you a sense of some of the factors that contribute to cancer disparities in the United States, and also to give you a sense of maybe where some of the main disparities are. And also just to say before starting that I don't have any specific conflicts of interest to disclose, and that the views presented here today are my own and not those of the US government. Okay, so that's essentially what I just said. Two things I want to address sort of from the beginning. For most of the time when I go through the talk, I'll talk about population differences or racial differences, but obviously both race and ethnicity are very important when you discuss things in the context of cancer health disparities. So again, just for anyone who's not immediately in the field, race refers to biological differences between groups that are assumed to have different biogeographical ancestries or genetic makeup, whereas ethnicity is more of a multidimensional construct. And here this often reflects geographic origins, historical differences, shared customs, beliefs, and traditions, and that may or may not have a genetic origin. Both of these factors are very important in not just trying to understand the causes of disparities, but also trying to figure out ways to overcome them. And in various studies that I'll describe, either race or ethnicity is used. The NCI defines cancer health disparities as differences in the incidence, prevalence, mortality, and burden of cancer and adverse health outcomes that exist among specific populations in the United States. And we've noted now for a long time that African Americans have the highest death rates from all cancer sites combined, and specifically malignancies of the lung, colon, rectum, breast, prostate, and cervix of all racial groups in the United States. So it's quite a significant health burden and disparity. So this is data from an old paper, but essentially it makes the point that the life expectancy for Black men and Black women is significantly shorter than it is for Europeans. So for men on average, there's a difference of about 6.5 years in life expectancy compared to European men or European American men. And for Black women, it's about 5 years shorter. And that's a significant amount of life years lost. And that's based on all-cause. This isn't just cancer, it's all-cause mortality. Now, when you look at the main contributing factors that do contribute to this specific difference in life expectancy, cardiovascular events in both men and women is a very significant factor. But as you can see here shown in the pink color, cancer is also a very strong and significant factor to the differences in life expectancy for both men and women in the United States. I mentioned this a little bit earlier, but when you look at all cancers combined, for both men and for women, African Americans have the highest lung cancer incidence compared with all other racial and ethnic groups in the United States. And you can see this for both men and for women. And what we're going to try and go through today is talk to you a little bit about some of the factors that contribute to these disparities. The other thing when we think about it is that it's a multidimensional issue. And one of them is that it's not just the increased incidence that we see amongst African Americans, it's also there's an earlier age of diagnosis. So this is a graph, and any data to the right is showing a later age of diagnosis amongst African Americans, whereas the lines to the left show an earlier age of diagnosis. And for some of these, including Kaposi's sarcoma and also non-Hodgkin's lymphoma, we're talking about a 5 or 10 years on average early age of diagnosis, and that is also quite significant. So you might be wondering, well, how does this happen? So this is, what I'm going to go through here are some examples of factors that can contribute to this, but it by certainly means not an exhaustive list. So one of the factors is something that we call etiologic heterogeneity. So what does that mean? That essentially means that any etiological factor, or any exposure that's associated with the disease, it could differ across populations and therefore cause cancer at different ages. So as most of you know, for most cancers, there is no one single etiological agent that's associated with the development of that disease. There are many. And individuals at different times in their life course could be exposed to different levels of that particular exposure. And I'll give you one example in terms of tobacco, because it's sort of related to the second point, which is, in general, African Americans start smoking cigarettes later in life compared with European Americans. It's about 3 or 4 age difference. And so when you consider the cumulative nature of that exposure, you might expect to see a different age of onset in terms of disease. The other, of course, factor that really impacts incidence, or the timing of incidence, is the access to early detection. So the more likely that you are to engage or have the ability to engage in screening activities, the sooner or the earlier age that you might actually end up with a cancer diagnosis. So these are all factors some factors which could explain the younger age of diagnosis. And one which I haven't mentioned here, but we'll talk about a little bit later, is also, of course, biology. Many of you may have seen in the news of late a lot of evidence talking about the increase in the rise of early onset colorectal cancer amongst the community. And to really address some of these early onset malignancies, the NCI has established the Early Onset Malignancy Initiative. I have written the link to it down here. But essentially, it's a national program which is really trying to understand why some of these cancers are occurring earlier, probe the epidemiology of it, the genomics of it, the biology of it, and also address, in this context, disparities. Because a lot of these cancers are, as I mentioned a couple of slides ago, are occurring in individuals, minority populations at an earlier age. One thing which we don't often think about or even talk about is, in addition to primary cancers, individuals who survive their initial cancer diagnosis are also often at risk of having a secondary cancer. There isn't a lot of work in this area, but I'll point you to one paper from 2011, which has shown that even in the cases, so these are all individuals, and this is a second cancer in those individuals. And it's shown for all sites combined, SIR is a standardized incidence rate, that for all cancer sites combined, there is a higher odds of a second cancer, or second cancer diagnosis, not just a primary cancer diagnosis, for many of these individuals compared with it's higher than whites. So when we talk about disparities, we often talk about the occurrence of that first cancer, but more research is now showing that it's not just that first cancer, there's also evidence that the incidence of second cancers is also higher amongst minority populations. One thing that I wasn't hugely aware of before I moved to the United States, I'm from Ireland originally, so it's kind of a small country, but geographical factors also significantly contribute to cancer health disparities. So this is just a graph of the United States. This is for everybody, so this isn't broken down by population. But what it shows is that if you look across the United States, just at all cancers combined, you can see a lot of heterogeneity and differences state by state to the incidence of cancer. So some of the highest incidence of cancer are found in the states of Kentucky, Rhode Island, Delaware, Louisiana, and New Jersey. You can see some of those up here. Whereas the lighter colors, you can see some of the lowest rates of lung cancer. So what this tells us, I should say all cancer. I work on lung cancer, so that's why I often say lung cancer when I mean all cancer, so I apologize for that. But in general, for all cancers combined, you can see significant geographic heterogeneity. Now, there are many factors that can contribute to this, but one of the most important factors is rural versus urban status. So this is a paper from, or a CDC report, I should say, from 2017, and it exemplifies this just a little bit. So I'm going to take you through it for some of the cancers. So in the colors at the bottom, you have the county that you live in. So this is where you actually reside, is classified as either non-metropolitan rural, non-metropolitan urban, metropolitan with less than 1 million people, and then again, increasing again in density, metropolitan with more than 1 million people. So as you go from light to brown, you have an increasing density of the population. Over here, what you can see, this is showing incidence, and this is showing the death rate. But as you can see here, as you go from a rural area to an urban area of residence, you have a lower lung cancer incidence and lower lung cancer mortality as well. And you can see for lung, colon, female breast is the opposite, but for cervix, for many of these cancers, the incidence and the death rate from these cancers is highest in rural areas. Obviously, as I said, breast cancer is the one that's a little bit different, but for many cancer types, we really do see in this direction very significant disparities. So what could drive that? Well, again, it's a complex factor, but I just want to mention and give you a few things to think about. One of them is that in many cases, it's been shown that a low socioeconomic status or a neighborhood can confer additional incidence and mortality for cancer risk beyond an individual's socioeconomic status. So what does that mean? It means that by geography, in some cases, there's an unequal burden of pollution. And also in some cases, there is decreased, such as in rural areas, decreased access to preventative services. Preventative services could mean tobacco cessation programs, efforts to help you give up smoking, but also in terms of screening. Because as you can imagine, screening centres and access to screening centres and the ability for one to get to a screening centre, be it in an urban area, if there isn't suitable public transport available, can be a difficult thing to do. And these are real factors that contribute, not just the stage at which a cancer is diagnosed, but an individual's likelihood to develop that disease. And it's also been shown in some studies that rural populations are more likely to have an unequal burden of pollution that I had mentioned, more likely to forego sometimes medical care and prescriptions due to cost. This is an economic issue, and also to experience things like psychosocial distress. I won't necessarily go through that, but you can read through it. But the point is that in the recent number of years, essentially, the community at large have documented these disparities in greater detail and come to recognize that it is a significant factor that contributes to disparities. A lot of it does have to do with access to care. So as I speak about a little bit on this slide, access to care, but really access to prevention mechanisms. So we talked about tobacco cessation, but access to vaccination for HBV and for HPV, and also access to screening, and trying to come up with ways where we can increase those tools and those preventative tools into the rural community. And sometimes you need innovative ways to try and do that. So we've talked about differences in cancer incidence and how that can vary by minority and population groups here in the US. But as I mentioned too at the beginning, there's also significant disparities in terms of survival. So as we'd mentioned, African Americans have some of the highest rates of cancer-specific mortality. And it's important to say that, as you know, oftentimes we take these measures, it's at a specific time point. It's in 2016 or it's in 2008. But work has also been done to track these trends over time. And for some of these cancer types, the magnitude of that difference in disparity, it is decreasing. But for others, it's increasing. And there are areas specifically that we have to pay attention to. When you compare African Americans with European Americans, some of the biggest differences in mortality are seen for breast cancer and also for prostate cancer. And that's shown here in these arrows out to the right. If you compare Asian populations with European Americans, you can see that they generally have a better outcome for all of these cancer types. And for Hispanic populations compared with European Americans, it's generally mixed and cancer type specific. So why are there these differences in mortality? So again, it's not that it's one specific thing. And so what I'm going to try and take you through are some examples of the factors that contribute to this. One of the obvious ones, we've touched on it a little bit, is the stage at which you diagnose a tumor or a cancer is very tightly related to the likelihood that, well, first of all, it increases the options of treatment that you can get. And in doing so, it increases the likelihood that you'll have a favorable response to that treatment. But in generally what we find, so the blue here shown is African Americans. And what you can find is that in general, they're less likely to be diagnosed with early stage disease and more likely to be diagnosed with late stage disease. So this, without question, is one factor that does contribute to the differences in survival between European Americans and African Americans, and indeed other populations in the United States. So we've mentioned this a little bit, but one of the issues that drives the differences in the stage at which a cancer is diagnosed is access to screening. We talked before about rural-urban differences in access to screening, but also there obviously can be others. But it's important to say that the uptake of screening for some cancers can vary. So in breast cancer, mammography use they found that the use is similar in an equal access to care setting. So that suggests that access to care is important. However, they've also shown for colorectal cancer screening that the uptake is lower among African Americans, even in an equal access to care setting. What drives that? We don't really know. There's also evidence to show that lung cancer screening at the moment, or the uptake of screening, is similar or maybe a little bit lower among African Americans. Something that we'll talk a little bit about later is that it's not just whether one willingly, if you're within a certain screening criteria, it's not about whether you willingly decide, yes, I will go for screening, or no, that I won't, in terms of population differences. One thing that we have found for lung cancer is that the criteria themselves can determine whether or not a population is likely to get screened. And I'll give you one example here, as I mentioned, from lung. So for lung cancer screening, one of the greatest breakthroughs that we've had, in addition to immunotherapy in the last number of years, is low-dose CT screening. So this is a spiral chest X-ray, which is recommended for high-risk smokers every year. So it's annual screening. The criteria for screening are age, so 55 to 74, your smoking history, so you must have 30 pack years of smoking. So that's actually quite high. So that's the same as smoking a pack a day for 30 years. It's quite high. And you must also have, if you are a former smoker, you can't have quit for more than 15 years. So they're the current recommended criteria for screening. So if you look at those criteria across the population and say, are there any population differences? Well, there are. So even though African Americans, it's a bit of a paradox, but even though African Americans are more likely to get lung cancer, their pack years is lower, so they're more likely to have a lower pack year history, and they're also diagnosed at an earlier age. So what this means is that the criteria that you use for the population at large are less likely to be applicable to African Americans. So what does that do? If we don't adjust it going forward, what it will do is it will increase the likelihood that you exclude African Americans from screening. And if you do that, of course, it increases the likelihood that you'll have a higher proportion of late-stage tumors diagnosed in that population. So even though we have something that's as good and as wonderful as low-dose CT screening, there is the potential that with the current screening criteria, we could exacerbate the disparities, and that's an important point. And this paper is a follow-up paper to our one, and it essentially shows what I've just said, which is if you look at the the eligibility, African Americans are less likely to fall within the age criteria. They're more likely to fall within the age criteria, and they're also more likely, as I mentioned, to have this lower pack years. So it makes them more likely to be ineligible for screening, and that's a problem. This type of this aspect of screening and eligibility isn't unique to lung cancer. Breast cancer, prostate cancer, colorectal cancer. There's many issues about screening guidelines and their ability to not discriminate, but to be applicable to various different populations. And so there are many conversations ongoing about how we can take evidence like this and incorporate it into screening guidelines. They haven't changed yet, but I think the evidence that's being generated certainly would be supportive of generating new guidelines. But it's important to say that mortality differences also exist even when there is no validated screening mortality. So it could be that access to screening and uptake in screening could lead to a difference in the stage at which the tumor is diagnosed across populations, and that could then impact mortality. But there are several cancer types like liver and esophagus where we don't have a screening modality, which we do also see differences in survival. And one of the key issues is access to care and also access to good quality health care, and we'll talk about this in a moment. This is another graph of the United States, and it's color-coded by the number of oncologists per 100,000 residents in the United States. And you can see there are some regions of the US where there is probably like DC region here, where there's very dense access and high access to oncologists. But this yellow here, this is zero. So for all the people who live in these areas, they have no access or very little access to oncologists. And that type of access or lack of access to care has a very significant impact on patient outcomes. Another aspect that has a very strong factor on outcomes is health insurance, right? And this won't be a surprise, I'm sure, to many of you, but this is just a few selected cancers showing the survival differences for breast, cervical, colorectal, and lung cancer by your insurance status. That's the only factor that's considered in this graph. And what it shows is that those individuals with private health insurance do considerably better than those that are either on Medicaid or uninsured. And this has been shown time and time again. And to make the point even again, there's many studies that show for many, not all, but for many cancer types, that within an equal access to care setting, such as a military health system, such as the one across our campus here in Walter Reed, where this study was done, lung cancer survival, where there are disparities, in general, African Americans do worse than European Americans. If you just take individuals within an equal access to care setting, where everybody has access to the same care, all those disparities go away. So what this tells you is that in terms of survival and mortality, access to care is extremely important. It's also important to say that a lot of the examples that I'm giving you are in the context of African Americans in general doing worse than European Americans, and that's because that's often the case. But there are a few examples of the opposite sort of trend, and one of those is multiple myeloma. This is a disease that has an identifiable pre-malignant stage, and we do know that there's an increased progression of that pre-malignant stage to malignancy amongst African Americans. But when you do have a frank cancer, what we see in multiple myeloma is that in general, African American patients have a better survival compared with European American patients, suggesting that there is, but not shown, that there is development of a maybe a less aggressive or more indolent form of the disease. It's important to say too, I mentioned this point earlier, but similar access to care alone does not guarantee equal access to quality care. So just because you have access to care doesn't mean you have access to high quality health care. So what does this mean? It means that in a regular medical setting, studies show that racial disparity in specialist consultations, as well as subsequent treatment with multimodality therapies for metastatic cancer exist. So these are just two examples. So European American populations are shown in black, and they're shown in black, and European American patients are shown in blue. And what you can see is that the consultation for surgery, chemotherapy, or radiotherapy was significantly lower in the same institution for African Americans as it was compared with European Americans. And the same thing here is in what treatment is offered. There's also significant differences. And this has been shown in many different settings and in many different cancer types. This is a paper that came out last year, and I like it a lot. I like the way they presented the data because I think it helps to bring the message a little bit clearer, especially too in terms of most people who work in this field are trying to think of, okay, these are the disparities, let's document them, now let's think about a way to diminish them or to make them less apparent. And what they did was essentially, I've shown here for breast and prostate, but they did it for many different cancer types. The solid gray line here that's shown is essentially the non-Hispanic white population. The blue line here is the non-Hispanic black population, and then there's various other populations shown here. But what they've done is they start out and they say, okay, what's the disparity between, in this case, survival between African Americans and European Americans? And so at baseline, it's about 2. So there's 2 times the likelihood that an individual with a breast cancer diagnosis would die compared to a European American. What they do then is they subtract the contribution of each of these factors to that specific disparity. And what they show is, if you consider stage, so they say, okay, let's just pretend everybody was diagnosed at the same stage. Now, how much of a disparity is there? And as you can see, it drops down to about 1.7. So it's still significantly higher. But you can see, and you see this in the figure for many other cancer types, the stage at which a tumor is diagnosed is amongst the biggest contributors to disparities in survival for multiple cancer types. Whether or not, for breast cancer, whether or not they get surgery is very important. Socioeconomic status is important. And as you can see, as you go along the line and account for all these other factors, a lot of the differences go away, but they don't go away entirely for breast cancer. For prostate cancer, it looks like they do. And again, is another way of driving home the same point that access to care and the uptake of care matters. What this graph also shows, quite interestingly, is the influence of neighborhood and socioeconomic factors and general social determinants, such as support mechanisms, which also contribute to disparities. This is another example from lung cancer. It's from a couple of years ago, but again, also shows the point that even among those with Medicare, so again, similar access to care, African Americans were less likely to receive treatment for lung cancer. And also in the case of renal cancer patients, they were less likely to receive surgical treatment. So I mentioned a moment ago that in addition to colorectal cancer, we see this recurring theme across many cancer types. And this is an example of, again, just of colon cancer, just showing from a more recent paper that, again, in a setting of equal access to care, African Americans with colon cancer are less likely to receive surgery and chemotherapy compared with European Americans. And so here, any number that's below 1 means that they're less likely to receive it. And it's for men and women across all ages, and essentially across most tumor stages. So there are various factors that contribute to these differences, and for every cancer, there's no one type, and in many cases, it's individual specific, and it can also be physician specific. But the factors that contribute, that have been shown to contribute to some of these differences in the uptake of care or the offering of certain treatment modalities include personal beliefs and fear, which I can certainly understand. There's cultural factors. There's also the patient-doctor relationship. There's patient bias, provider bias, and even just the communication between a doctor and the patient is also extremely important. And the way in which treatment options are communicated has a significant impact on outcomes. And something which we've also seen in some of our studies is also comorbid conditions. So on one of the studies earlier, we had shown how in some cases the likelihood to have surgery or to have chemotherapy was less in one population compared with another. Well, your physical fitness or your ability to be a candidate for surgery or to be a candidate for a really intensive and grueling regime on chemotherapy may also be related to any comorbid factors that you may have. So in lung cancer, for example, some of the ones we think of are COPD and emphysema, which if you have them, don't often make you a good candidate for a lot of the therapeutic options that are available to you. So these are all factors that can influence the uptake of care among cancer patients. So for some cancers, as I think we've shown, disparities persist even in settings of equal access to equal care. Sometimes that's driven by equal access to unequal care options, but this is an example here of survival. It's an old graph, but I show it because it's one of the first to show this point. So this is the survival curve for European Americans and African Americans. And as you can see, African Americans have a worse outcome. This is for breast cancer. And this is in a clinical trial setting. So everybody has the same access to care. They're all getting the same drug. Everything is very well controlled. So what's going on? This is also a study from just very recently, actually, colleagues again who work in the area. But they also indicate here that the time to breast cancer surgery was delayed for non-Hispanic Blacks compared to non-Hispanic white women within the military healthcare system. So again, this is one of these equal access care settings. But the racial differences in time to surgery, so it did exist, there was a disparity, but the differences did not account for the observed disparity that there was in survival. So they accounted for it in the analysis, and they said there's still something else going on here. And again, this is an example of a clinical trial setting where, again, everybody has the same access to care. This is breast cancer, premenopausal, postmenopausal, and the ones in red are where there's a significant disparity. And so what it shows is that even in settings where people do get the same access to care, the same uptake of care, everything's accounted and everything is controlled for, there can still be differences in survival. And so what that leads you to ask is whether or not biology could be a contributing factor. And essentially the answer to the question is that it is a contributing factor. It doesn't explain all the disparities. Even if you addressed every difference in biology that there may be, it's not going to counteract all the disparities that we see and that we encounter. What I wanted to do was to spend the second part of the talk going through some of the examples of where we have discovered differences in biology and what that could mean. So if one of the first things that we know, and I'm going to jump from cancer type to cancer type again, just to make some key points as we go through. We do know, and we have known for quite a while, that African American men are more likely to be diagnosed with aggressive prostate cancer. So one of the ways in which prostate cancer aggressiveness is measured is on something called a Gleason scale or a Gleason score. And the higher it is, the worse, the more aggressive the tumor is. And what you can see here is that, so this Gleason 7 and greater than 7 is, greater than 7 is the higher score. The blue is the lower score. And you can see that African American men are more likely to have a higher score than European American men. And this is a feature of aggressiveness that's used extensively in the medical field. Some work has shown that there is a role for genetic susceptibility in explaining some of the disparities for African American men. Many of you will have heard of this region on the chromosome at HQ24. So MYC is there, but there's also some other important things there. But people have been doing things called GWAS studies for a long time. I think there's some lectures on GWAS studies later on. But essentially what it does is it looks across your entire genome, so each of us, our normal genome, and looks for things called SNPs, which are just single nucleotide polymorphisms. And we all have them. It's what drives the heterogeneity between all of us. But some of these can be associated with an increased risk for disease. So the inherited BRCA mutations that many of you will have heard of, they're examples of SNPs that are associated with an increased susceptibility for disease. And what they have found is that there is this region on AQ24 that is associated with increased risk of prostate cancer. But what they've subsequently shown is that this is the specific region that's associated with risk. There's an excess of African ancestry at that region. So essentially what they've shown is that that region that's associated with risk is more common amongst men with African ancestry. And they estimate that about 50% of the disparity that we see in terms of incidence, I'm only here now talking about incidence, can be explained by that specific region. And there's been some other regions, including some on chromosome 17, that they say maybe contribute to about 10% more. So this is an example of where there seems to be some evidence for biology driving the increased incidence of a certain type of cancer. And probably prostate cancer is one of the best examples of that at the moment. But the same locus that I was talking about has not just been associated with more or higher incidence of prostate cancer, it's also been shown to be associated with more aggressive prostate cancers. So that same locus that I talked to you about, the one that's associated with a higher incidence of disease, they've also looked at whether it's associated with a higher Gleason score and metastasis, and they see in both cases that it is. And this is evidence that's not just associated with more or higher incidence of disease, but incidence of a more aggressive disease. Another interesting example from Jun Yang at St. Jude's in Tennessee is he's shown that there's an increasing proportion of Native American ancestry that's associated with an increased risk of childhood ALL. So a lot of what we've talked about in this so far has been specifically looking at differences in European Americans and African Americans. But there are many minority and underrepresented populations in the United States, and Native Americans and American Indians are one of those. So this was a very interesting study, and it has a couple of implications. One of them, as you might imagine, given what we talked about earlier, is related to screening. So if you have an individual of a certain degree of ancestry, which is a higher susceptibility to disease, then that's an individual that you might target for more intensive screening. But what the authors of this paper also found is that children with more than 10% Native American ancestry needed an additional round of chemotherapy in order to respond to the treatment. And they found this through a retrospective analysis when they went back and tried to look at why certain children responded and why some didn't. And this, of course, it can have screening implications, but it also has very practical therapeutic implications as well. And it tells us that, we talked about this at the very beginning of the talk, that ethnicity is important, but race and genetic ancestry are also important. And in any of these types of studies, we try and consider all of them as much as we can when we try and understand, or when we are trying to understand the specific problems at hand. Many of you will be familiar with, we talked about germline mutations such as BRCA. Many of you will be familiar with somatic mutations. And of course, these are what we know and talk about a lot in the context of driver genes and driver mutations. And a lot of precision medicine is geared at targeting these types of mutations. This is just one example from colon cancer by Gouda and colleagues. And in their paper, they found an increase in FRNA6 and FLCM mutations. Essentially, they only found them in African Americans. The studies that we have done in lung, I would say for the main part, and for most of the studies that are in the literature, what we find is that for a given cancer type, the main genes that are mutated are the main genes that are mutated. So P53, KRAS, STK11, in essence, the same genes are mutated, but every now and then, we do find specific genes where there's a higher mutation frequency in one population. This is important because it tells us, obviously, the mutation spectrum tells you about etiology. So in cases where we don't know the factors that are driving an increased incidence, it gives us a clue. The other thing is that, as you know, and we talked about, a lot of driver mutation genes are candidates for precision medicine. And so that's another example of why, or how, I should say, these types of findings can be taken further. And it's also too important in the context of tumor vaccines and drugs that are being targeted towards specific antigens. Because what we also see, and what's shown in this paper, is that even though the mutation frequency of this gene is higher, it's not just the frequency that's higher, the specific codons that are mutated are also different. And that has implications in the context of tumor vaccines. This is, again, is an example going back to prostate cancer. So some of the main somatic changes that are seen in prostate cancer include the temporous ERG gene. It's a fusion. P10 deletion is very common. SPINK1 and SPOP mutations are also very common. And essentially what's graphed here is the frequency of those genetic alterations in both African Americans and European Americans. What's very striking is that in general, there's a very high frequency of this fusion gene, the temporous ERG fusion gene, in populations of European descent. About 50% of prostate cancer patients will have that particular fusion gene. There's an intermediate frequency found in African American patients. It's only about 23 to 30%, and it actually is quite uncommon amongst Asian populations. So again, this is another very solid example of how specific somatic events can vary by population and ethnicity across the world. And this is important because this type of fusion gene is the driver. It shapes the biology of the tumor. Now, why it's different, I don't know. I don't think the field knows, but it does highlight population differences in biology and consequences for the tumor biology even of itself. As I mentioned up here as well, P10, which is a very common tumor suppressor in many cancer types, it's very common to have P10 loss in European Americans and African Americans, but quite uncommon to have it in Asian and African American patients. This is a paper that came out just towards the end of last year. It's from one of the TCGA consortia. And essentially, they looked at pan-cancer. So they took every cancer type that they had in TCGA, and if they had about, was it 10 or I think it was 10 or more individuals of African American ancestry, and they just compared pan-cancer, what the genomic alterations look like. It was kind of at a high level perspective. But essentially what they found was kind of two quite interesting things. The first, which isn't shown here, but in general, they find that there is a slight increase in P53 mutations among populations of African descent pan-cancer, and also that there's more genetic alterations in the PI3K pathway, again, a very oncogenic signaling pathway in cancer. This is across all cancer types. So again, it sort of suggests that there's a component or a contribution of genetic ancestry. The other thing that they found was that for many cancer types, and significantly for breast cancer, ovarian, and for head and neck cancer, that there was a higher degree of genomic instability in those tumors. And then for kidney cancers, they found that there was less. And this is both across the entire chromosome, focal regions, or whether it was just at the arm or chromosome level. We've talked a lot about germline somatic differences, but at the transcriptome, there are also significant differences by population. One of the best characterized of these is in breast cancer. This is an old paper, but it was one of the first that started to show this. So we know, in essence, that the triple negative form of breast cancer is more common in African Americans compared with European Americans, but it's also a lot more common in of women in Africa. But as you can see down here, as I mentioned, this is one of the first papers to suggest that there was this difference. And what they found was that the estrogen receptor, which is of course a component of the triple negative breast cancer, was less likely to be found amongst populations of, in this case, white compared with black. But this has been replicated multiple times since. This is important because in breast cancer, it could be one of the factors that contributes to the differences in outcomes, simply because for estrogen receptor positive breast cancer, you have a therapeutic option with tamoxifen. For HER2 positive breast cancer, you have options to treat those patients with Herceptin. But for triple negative breast cancer, there are very few effective therapeutic options. Obviously, PARP inhibitors are used, but there are very few that are effective and give you long-term remission for your disease. So this is a very well-characterized transcriptome-based molecular difference in tumor biology between European Americans and African Americans. But even more than that, as I've mentioned, it also extends to populations in Africa. It's a very specific subtype of breast cancer that you see more often among individuals of African ancestry. I put this slide in because I just find it interesting, kind of walk back a little bit what I just said, but it even has been shown, so triple negative breast cancer aside, then again, this is data from a clinical trial, but there are disparities in survival irrespective of the tumor subtype. All right, so if you try and again take a control for what subtype it is, for the ER positive disease and luminal A and luminal B, the the survival of women with this subtype of breast cancer is still worse amongst African Americans. So while some of what we understand in terms of triple negative disease may explain some things, it doesn't explain all. And that this is an example of a graph that highlights that. This is an example from lung cancer and some of the work that we have done. It's one of the first papers that we've done more since. But again, it supports what we've seen in colorectal cancer, in breast cancer, in colorectal, in endometrial, is that a lot of the tumor biology in it is shared between these populations. But in almost every tumor type that you look at, you will see a distinct section of biology that's different. For our study and for several other studies, we often find that some of the genes that are perturbed only amongst African Americans relate to stem cell signaling, invasion, metastasis, those types of pathways. And in our case, in European Americans, we find differences that primarily relate to cell proliferation, cell cycle, mitosis. That's interesting, as I said, because it gives us an understanding of the tumors themselves, but we've also been able to correlate those differences with differential response to therapy. And I think that is also important for potential translation. Also at the transcriptomic level, a colleague of mine, Stefan Ams, has shown that back again in prostate cancer, that there's an enrichment of an interferon, which is a type of inflammation, an interferon signature in prostate tumors from African American men. And the presence, he's since shown just recently, that the presence of this particular signature is related to a germline mutation. So that's also quite interesting work. But again, it reinforces the idea that you have transcriptomic-based differences in tumor biology. I know somebody might ask this question, but when we talk about the immune response, one of the most important things that we think about these days is response to immunotherapy. And do we know of any differences in population differences in response to immunotherapy? This paper was published online, so I don't have a full reference for you, but just a week or two ago. But it was the first study that was really powered to look at whether or not race and ancestry was related to response to an immunotherapy treatment. The reason for that is most of the other trials, the enrollment, they're often small trials, so maybe a couple of hundred people, and if they're in the US, the enrollment of minority populations is very low. There are many reasons for that, but the enrollment was quite low. But in this particular study, it was called the PROCEED trial, and it was the evaluation of this immunotherapy for asymptomatic or minimally symptomatic metastatic castration-resistant prostate cancers. And essentially what they found, over 10% of the individuals in this trial were African American, so they were specifically powered to look at it in their analysis. And race was a significant independent predictor of outcome, but it was a predictor of response. So it goes back to something I'd mentioned earlier. A lot of times what we talk about is an adverse response, or the less likelihood to respond to something, or the less likelihood to survive. But this is the first trial. It's a very specific form of immunotherapy. It's not PD-1 or PD-L1. This is actually a vaccine-based treatment. But essentially they found that race was an independent predictor. So being Black meant, or African American, meant that you were more likely to respond to this therapy. Why that is, we don't know. The paper doesn't really go into it at all. But it's an interesting observation, and B, it underscores the importance of looking at race and ethnicity and genetic ancestry in these studies. You'll think back to the example I gave you of acute lymphocytic leukemia and Native American ancestry also being associated with response. So there aren't very many studies that have looked at this in a good way, in an empowered way. But as you can see, there are examples of when it is looked at that it does turn out to be important. So in the interest of wrapping up and leaving time for questions, I just wanted to come back to this slide that I showed you at the beginning, which is essentially looking at or outlining the differences in life expectancy that exist. Not all of these differences in life expectancy are related to cancer, but a significant proportion are. During the lecture today, I've tried to highlight some of the factors that contribute to that. In some cases, it's factors that contribute to incidence. Those factors can be related to access to care, so access to screening. It can be related to exposure to various carcinogens or exposures, but it can also be related to the worse survival that many individuals in the United States who are in minority or underrepresented backgrounds experience. And that is also a very multifactorial process in and of itself. One factor more and more that we think is important, but certainly not the entire picture, is biology. And with the new technologies that are available to us, leveraging what's in TCGA, but even going beyond that and including very well annotated samples in our studies, we are beginning to see some aspects of biology that are different across populations. And really, for most of us in the field, we acknowledge that there are many factors that contribute. I think you hopefully kind of seen that from the talk today. Sometimes it involves the host, which refers to the biology that we talked about. The environment is important, socioeconomic status, geography that we talked about, whether you live in a rural area or an urban area, for many cancer types, has been shown to impact not just your incidence of cancer, but also your mortality. And also behavior. So whether you're likely to smoke, what your diet is, your beliefs, whether you're likely to if surgery is offered to you or chemotherapy is offered to you, whether you're likely to say yes or no, the reasons, the very complex reasons that go into those decisions, they also vary significantly, but they also are some of the factors that contribute to disparities. For those of us who work in the field, we often work in one of these little boxes, but it's very important for us. Sometimes we might work in one or two, but in general, it's very important that we think of the problem as complex as it actually is, and to work with colleagues and to talk with our colleagues who work in all these different areas. Because by doing that, we may think of factors that may impact, like biology, for example. We generally don't look at socioeconomic status or where the person is living, whether it's rural or whether it's not rural, but maybe we should. So we're now going back and we're trying to geocode our data to include those types of factors in our analysis. So the main point of this concluding slide is just to say that there are many key determinants of health disparities, even amongst the same cancer type, even amongst the same individual. And they can be different for both incidence and also survival. But it takes a comprehensive community to try and tackle them, and hopefully in the coming years to hopefully diminish them. So with that, I think we've left time for questions. If we don't have time for them today, I'm here on campus. I work in building 37, so feel free to come and knock on my door anytime. Thanks for your attention. Yes?
Thank you so much for the talk. It just reminded me, or let's say highlighted, the reason why, at this current time age with, these very much advanced options for therapy, all of that, and the reason why, this is the reason why cancer has not been eradicated, just because of the amount of disorders that apparently, according to you, are not quite understood by, factors. So thank you, that was quite interesting. I have two questions that are very, very short.
No problem, we have time.
The first one of your slides did mention that second primary cancers referred to are quite common in African Americans. The question is, secondary cancer, is it the same cell of origin as the primary cancer, or
no, different? Yes. Yes. A very unfortunate individual who has had a primary cancer diagnosis, survived, and then been diagnosed with a secondary cancer.
So they're relevant and independent of each other.
They seem to be, except that the treatment that you get for that primary cancer can be strongly associated with, like radiation treatment. Radiation is a carcinogenic agent. So there are some confounding factors, but essentially a second independent tumor.
And my second question, when you were talking about the multiple myeloma incidence,
Survival, yeah.
In European American versus African American, is that only specific for multiple myeloma or any of both?
So that one was multiple myeloma. And that was one of the examples where we see that opposite trend, where African American men and women tend to do better.
But would it be accurate to generalize that in other
No. But one of the things, so one of the reasons that I'm always very happy to come back and give this lecture every year, thank you, Terry, for that, is that before I came here, I never thought about my work in the context of disparities. I didn't, as I said, I came from Ireland. The notion of disparities was quite unknown to me. But it's because we're a very small, singular sort of population, and you can drive the whole length of the country in 4 hours. But it's that I would challenge each of you when you think about the work that you're doing. And I know it was very high level perspective of all the disparities that exist, but just think about it. Whether it's the aspect of biology, an aspect of treatment outcome, or something that you're doing, just keep that at the back of your mind, because it's a very significant public health problem that we haven't solved. But anything we can do to diminish the differences in life expectancy and the quality of life, I think, is important. So if you're doing a study and you have 200 tumors and you never thought before, well, where do they all come from? My advice to you would be to take a look and take a closer look and see if there are any differences by population. Yes.
I had a question about what's being done to address this by organizations with Paris in America. I was thinking about, like, I don't know if you know, in Liverpool, there was a Northwest Cancer, and Liverpool used to be like the capital of Europe, and they really targeted people in the community. So I was wondering what's being done here.
There's a lot, and I certainly won't pretend to be able to speak to it entirely. But the NCI, our director is Dr. Doug Lowy, and when he assumed that position earlier this year, he again highlighted cancer health disparities. It was one of the key areas that he was interested in tackling. So there's many different ways to do that. Some of it is through funding, obviously in extramural grants, but even intramurally, he's very focused on the cancer health disparity intramural research program as well. This slide here tells you that you don't just do one thing, right? So you have to do many different things. Yeah, and so oftentimes that sort of work is funded through NCI comprehensive cancer centers and through grants. But at the core, like whatever the problem is, so whatever the factors that's contributing to disparity, let's just say it's a low access to, or a low uptake of screening. So you have a modality that works, but populations aren't using it. You need to figure out why. Sometimes it's as simple as just, well, there is no bus and I don't own a car, so how am I supposed to get to the screening van? I can't do that. So then you could, you figure out what the problem is and then you try and fix it. Sometimes it's a case of, well, I don't trust that specific provider, or it could be that my uncle went to that hospital last year and he didn't do so well, so I'm not going back. So there's multiple factors, and this is just a very simplistic way of trying to address it. But a lot of the research that's done is trying to understand what are the factors, if it's behavioral or if it's socioeconomic, as it could be, and sometimes it's both, right? It's not just one. And then on the research side, obviously with the types of things I talked about towards the end in terms of biology, that in recent years, there's also been an uptick. So what I'd say at the NCI level, and certainly I could probably say at the NIH level too, there's a lot of funding on the ground for community organizations. A lot of that is administered through grants to try and have outreach, but really to also do the research to try and say, well, not that I know what the problem is, let me come and fix it, but figure out what it actually is. That's often focus groups, observational research, a lot of the typical epidemiological research that's done. And there's the NCORP, I'm not great at my acronyms, but one of them was up on the website. There's a network of those centers across the US who work together so that you don't sort of reinvent the wheel. So I'd say in the last 4 to 5 years, I have seen many advances. And also the key organizations, so AACR, ASCO, and all the key clinical research organizations in the US, they have highlighted disparities within their organizations of something that should be addressed. So some of it is top down, like this is a problem, you should be aware of it, think about it and what you're doing. And other times it's more on the ground. But I would say I have seen more of that high level highlighting of the disparities problem. Even the last 2 years, it's been even more. And I think that's a good thing. Yeah.
Have you seen differences in anatomical location of these different cancers?
That's a good question. So it's at the hand wave, the answer is yes and no. But I will speak specifically on lung cancer because that's the cancer that I know the most about. Lung cancer is primarily non-small cell and small cell. And within non-small cell, you mainly have adenocarcinoma and squamous cell carcinoma. Surprisingly, there is no real difference in the anatomical. So adeno is mainly distal and squamous is normally proximal. There is no difference in the histological subtypes for lung, even though I often thought there would be because the pattern of smoking is so different between the two populations, but there doesn't seem to be. But for other cancer types, there are, and that often helps you go back to learn about etiology and things like that. I think colorectal cancer is one where we have seen differences, especially the early onset, actually. Okay, all right. Well, thank you very much for your attention today and enjoy the rest of the course.
Today we have Jill Smith giving the second lecture. She got her MD degree in Florida and then did a residency in Missouri. And she became an assistant professor at Penn State in 1987 and subsequently advanced and was promoted to professor. And then in 2012, she actually came to NIH and worked at NIDDK for a few years, Director of Clinical and Translational Research. And then she moved to Georgetown. She's a professor of medicine there. And her talk today is Translational Research, Bench to Bedside Clinical Trials. Jill.
Okay, thank you. Can you all hear me in this thing here? Okay, good. Okay. Welcome. So you're not supposed to say what year we got our degrees because then everybody figures out how old you are. But I'm like an endangered species because there's very few people out there that are clinicians, scientists that, you know, actually still do the clinic and still do the science. And so, you know, I like to think I'm one of those, you know, dinosaurs or whatever, but there's very few out there. But I like that because I have a basic science research lab. And when we have things that we find that work, I can take it to the clinic because I have my medical degree. And then the other thing is it works the other way around. I'm in the clinic and I find problems that patients have, and I say, we need a solution for that. And then we go back to the bench and we try to come up with a solution for the problem so that it works both ways. So yes, I teach at Georgetown and I have a lab there and I see patients. So I have to figure out how to work this thing. Is that, oh, that's the pointer. Aha, okay. I have some disclosures. I have some patents. And you'll learn as you move on in your careers that you have to always do these conflict of interest forms. The NIH likes to do those. So, so today we're going to discuss how to understand taking an idea that you have from the research lab to patient care. What are the steps that are involved in conducting clinical translational research trials? And how to understand overcoming some of the obstacles that we have in drug development. And then I'm going to give you an example of one of the projects I'm working on right now. And then the pitfalls and the prizes that we have with this. So, so first of all, when we talk about going from working preclinical research and how many of those brilliant ideas, you know, we all have the most brilliant idea. And we're all going to get the Nobel Prize, right? So, but that the reality of it is that, you know, of all the brilliant ideas, and don't get me wrong, we all have brilliant ideas, and I like to think that my ideas are brilliant, your ideas are brilliant. But how, in the long run, how many of those great discoveries that happen here in the preclinic and in the bench are actually going to make it out to patient care? And this is the National Institute of Health. We're not the National Science Foundation. So, you know, we're interested in patient health and patient care. So your research is supposed to be translational, and your research is supposed to be about human subjects. So, but unfortunately, you know, many of our brilliant ideas don't make it out there into the clinic. But that's supposed to be the ultimate goal. And then I found this paper about the overview about drug development process. And I'm just really shocked at the expense of each of these different phases. And we'll talk about phase 1, 2, 3, and how much it costs to actually take a drug from bench to bedside. I just think that that's amazing. And I think actually a lot of these costs are overinflated. Because I can tell you I've done many of these on a shoestring budget, so they are possible, but a lot of it, industry does overinflate the costs. But it does take time, and so, you know, it does take, you know, years to do this. So the drug development, the average time in the United States is about 12 years for an experimental drug to go from the laboratory to the medicine cabinet. So, and it says here only 5 out of 5,000 drugs that enter preclinical testing actually move on to human testing. So that's kind of amazing. And then one of these 5 drugs that are tested in people is actually approved. So that's your bottleneck there. then the process of getting a drug approved here in the United States is that it has to go through the governmental regulatory agency, and here it's the Food and Drug Administration, the FDA, and they control which drugs and the regulation of them and which drugs can actually get approved. So most of you here, I presume, how many of you are like doing preclinical studies? Anybody here? So yeah, okay. So that's where most people are who are in research, doing a lot of preclinical studies, and you're working in the lab. And of course, this is very important because, you know, you don't want to give a compound to a human being that is going to kill a mouse. You know, so you have to test it first in the animals and make sure that it's safe and that it works, and also in cell culture. So these testings are very important. And as a researcher and as a mentor, I just want to throw out there to all of you that the integrity of science is critical at this point, because there are students that are trying to get their thesis done and they want the data to work, and sometimes it doesn't work. You can't fudge the data, and that's so important, because this is the most important stage. And if that p-value comes out to be 0.06 instead of 0.05, that's the way it is. But that's important, because if the data is fudged in any way to make the statistics look better, you could kill somebody if this gets into the clinic. So, you know, I publish papers that sometimes say the p-value is 0.06, because that's the way it is. And, you know, it may mean that you need a higher N, or you need to go back and try a different animal model, but that's okay. It's okay to report the data as it is. And it's very important not to try to twist the data in a way so that you get your paper published or your thesis accomplished on time, or to please your mentor, or whatever the reason, or to get your promotion. So I just put in the plug for, you know, integrity in science. It is so critical. And in the long run, it's only going to hurt other people if people try to twist the results. So it's beneficial. We all want our drugs to be successful, but the honesty is the best thing, and that's what's going to move the drug forward in the long run. So this is where most of us are, and I have a basic science lab, and I had to inject my mice today with gemcitabine. So I mean, I'm there too in different projects. So then when we go from animals to humans, the first phase that we go through is what we call the phase 1 trial. And the phase 1 trial doesn't have to include a lot of people. In some situations, it can even be normal controls. However, I will say that, you know, obviously in oncology, you want to test it in cancer patients. You're not going to give a chemotherapy agent to normal controls. And I'm getting ready to start a trial right now, and you want to kind of test the drug in the population that you're going to be testing it in is better, but it's not required. And the other thing about a phase 1 is, you know, you're doing it to figure out what's the best dose and is it safe. Sometimes we want to do the pharmacokinetics because this drug's never been tested in humans, and you don't know if it gets stored in fatty tissue, if it's excreted in the kidneys or whatever. But the important thing about the phase 1 is it doesn't matter if it works, because that's not what the phase 1 is about. So you don't have to compare it to anything. You don't have to show that it's shrinking your tumor or anything. You just need to figure out the dose. That's what's important in a phase 1 trial. What dose is the best? Sometimes what's the best route of administration? Are you going to give it sub-Q or IV or PO? And the pharmacokinetics. So then the phase 2 trial is looking for efficacy. And that's where you got to show that your compound is better than a placebo or better than the standard of care, because we usually don't give placebos to our cancer patients. Most IRBs won't allow that. And it still doesn't have to have a whole lot of people. Sometimes it can have less than 100 patients. But you have to have a primary endpoint, and I'll get to that in a minute. And it has to be, usually it's blinded. That means that, you know, the person who's doing the study doesn't know and the patient doesn't know what they're getting. But the important thing is you want to know, does this compound work? Is it more effective than a placebo? So that's the main part about the efficacy, and I'll get into randomized controlled trials. So then the phase 3 trial is usually a lot more people, and typically you're comparing it to the standard of care. There can be several different groups. You can have several different arms of this study, but it's important that there's an equal chance of being balanced between the groups. And you want to determine how this new treatment compares to either placebo or mostly standard of care. And then there are what we call superiority trials and non-inferiority trials. So why would somebody want to show that their drug is just as good as what's already out there on the market? Well, if their drug happens to be less expensive, that may be one reason why they could get it out there. Maybe it works just as well as what's out there, or maybe it has fewer side effects. You know, years ago, gemcitabine was approved to treat pancreatic cancer over 5-FU only because it had fewer side effects. It didn't improve the overall survival, but that's why it was approved by the FDA. So it was a non-inferiority trial that got gemcitabine approved. So then there's the phase 4 trials, and these are typically the trials that occur post-marketing. And for example, hepatitis B vaccinations, they had large phase 4 trials to see now this vaccination is approved. We're going to test and see, was it effective in large populations of people around the world globally? Does it prevent hepatitis B? And so that was taken into thousands of people, many, you know, globally, and that's post-marketing. So that's what a phase 4 trial is. So sometimes you'll want to do a pilot study. And, you know, for many of the young investigators here, there's all these little pilot grants you can get to test your idea and see if it's going to work. Well, the reason for doing that is, and usually it's open label, you're going to take your compound and you're going to give it to some people and see if you see some effect. The reason why you want to do that is to kind of get a tentative response rate so that you can calculate your sample size for moving forward. Because a biostatistician is going to ask you when you're trying to figure out your sample size, well, how well does this work? And you can tell them how it works in your mice, but you know, you really don't know how it works in your humans. So one time I did a pilot study and had about 25 people in it, and we figured out how the drug was working. And then when we figured out what percentage of the people seemed to respond to the treatment in an open label fashion, then we calculated our sample size to go into the phase 2 trial. So that's one reason you do a pilot study. So what are randomized, usually controlled clinical trials? And that's that you have an equal chance of being assigned to one arm or the other. And one little caveat here is that it's important if you're designing your own randomized clinical trial that you look at what your primary endpoint is going to be. And whatever that is, you should stratify your patients based upon your primary outcome. I did a Crohn's study one time, and our primary outcome was going to be improvement in the Crohn's disease activity index score. But we didn't randomize patients whether their score, they had to have a certain score to get into the study, but we didn't randomize them so that they were actually balanced. And it turned out that the people that had higher scores and worse disease got randomized to my treatment compared, you know, to the placebo. And fortunately, in the end, the treatment worked so well that you know, it didn't matter. But if it had been, you know, borderline, you would have wanted that to be the same at the beginning and to show that they dropped more in one than the other. So when you're doing a randomized trial, whatever your primary endpoint is going to be, if it's going to be, your RECIST criteria that you're going to shrink this tumor by a certain percentage, then you have to make sure that you randomize people at the onset so that their tumors are equal size. That's the sort of thing. So, so that's how, and it's important because you want to make sure that you have equal distribution in case there's differences, stage of disease, metastases. You want to make sure it's balanced because those things, as we learned in the last talk, they all affect response and mortality. So how are patients' rights protected as you're moving into the clinic? And this is very important because there's legal codes that govern how we do clinical trials. The important thing is that you have to have informed consent. And informed consent doesn't mean that you just tell the patient what you're going to do. They have to truly understand, and you have to go through the consent form process with them. And then there are different scientific reviews. Now, over at Georgetown, we have the Lombardi Cancer Center. They have a scientific review first for cancer studies before it goes to our IRB. And then the IRB has a panel of people that will review your application and tell you if it's okay and you can treat patients or not. So it's mainly for the safety of patients. Once you start the trial, then you have a data safety monitoring board. And this data safety monitoring board will actually periodically review your data. And they probably know, typically, what group A is and what group B is. Group A is placebo, group B is getting active drug, even though the investigator doesn't know. And you'll have early stopping rules. For example, if everybody who is in placebo is dying and everybody in your investigational drug is doing well, the Data Safety Monitoring Board will have the right to stop the trial earlier and no longer enroll people into the placebo arm because it's for the benefit of the patients. So, and the investigators won't know who's getting what, but the Data Safety Monitoring Board will know which group they're in. And the other thing is, we learned a lot just now about BRCA testing and SNPs and things like that. If you are collecting blood samples from any patients in a clinical trial and you're going to store their blood and you may want to use it in the future for a biomarker to see if they have a higher risk of getting this sort of cancer or if there's a predisposition, you have to have their consent for genetic testing because it could affect them getting life insurance. And so if you find that they have some SNP that predisposes to getting breast cancer or lung cancer, and that information becomes available, they have to have signed a consent saying it's okay to test for that. Now, most patients don't care, but they need to know that that's a true possibility because insurance companies can take that information and turn it around and not offer insurance if they're in a high risk. So now, what do you do before you can even start the clinical trials? Well, one of the first steps is that you have to apply to the Food and Drug Administration to get an investigational new drug or an IND. And even if it's an old drug that is out there on the market and you're trying to use it for a new indication or at a new dose, if it is different than what the FDA approval dose or indication is, you must apply for an IND. That's it. So, if you're going to take some medicine, and for example, I used low-dose naltrexone in our Crohn's patients. Naltrexone's an old drug, it's been around a long time, but it was approved at a 50 milligram dose to treat alcohol withdrawal. We were using it at 4.5 milligrams to treat Crohn's disease. It was a new indication. It was a new dose. I had to apply to the FDA to get an IND number in order to treat people, even though you're repurposing an old drug. If it's not the indication that it was approved for by the FDA, you still have to get an IND. And the IRB will not approve your application for your IRB protocol unless you have approval from the FDA for your IND number. Now, when you submit your information to the FDA, there's a couple forms that you need to fill out, the 1571 and the 1572. And when you submit all your information to the FDA has to reply to you within 30 days. If they don't, your number's automatically activated. So usually on day 29, I get a call from the FDA and they're responding to you because they know that the clock's ticking. And they'll send you some letter or something saying, we've received your application. You know, you cannot treat patients until da-da-da-da-da. But they have to respond to you within 30 days. So the information that you have to put in your IND application is you got to include the results from your animal experiments or your mouse, the cell culture experiments. You have to say how, where, and by whom the new studies are going to be conducted. Is it going to be a single center? Are you going to have a multi-site study? And then you have to give them information about the compound, even if it's already an FDA-approved drug. You got to give them the CAS number. You got to give them the chemical compound structure. And they want to know the pharmacokinetics of the drug. They want to know everything about the drug. And then if possible, say you're trying it for a new indication, what's the mechanism of action? So this is why, getting back to your animal studies, you have to figure out how is this drug working in this new condition. And because this is all information you have to provide to the FDA for them to give you an IND number. So what's the 1571 and 1572 forms? And I know you can't read that, but the 1571 form is all about the sponsor and the drug. So for example, one that I just got from the FDA, I am the sponsor. If you're working with a pharmaceutical company and like Merck or somebody, usually they are the sponsor of the IND, meaning that they're responsible for the studies and everything else. But if you're going bench to bedside and you're the investigator, you are the sponsor of your IND. Now, the reason that's important, not only are you responsible for everything about the study with the Food and Drug Administration, but each year, whether you enroll any patients or not, you have to submit an annual report to the FDA. And even if you don't have any patients, the study's not up and going, and you say no patients this year, you have to send them something. Otherwise, they can fine you $10,000. So it's very important when you're your own sponsor that you do your annual reports on time. And every time you submit something to the FDA, if they tell you they want you to make a change in your protocol, or they send you a letter and they want you to respond, you have to fill out one of these 1571 forms. You always put your IND number here so they know who's in charge of it. And then every time you send in a letter, you change the serial number. So with your initial application, the serial number is 0. And then the next time you write back to them and say, okay, I've revised my protocol, this is serial number 1. So that's where you are with the 1571. With the 1572 form, that's information about the investigator and the site. So they need copies of the clinicians who are going to be running the study. It has to be their full curriculum vitae, not a 5-page biosketch, and it has to be hand-signed and dated, and they want a paper copy of it at the FDA. So you have to do that. And it talks about the site. You have to talk about, you know, who's the lab where you're going to be drawing the blood. Are they licensed, the investigational pharmacy? Are the pharmacists licensed and up to date? And you have to submit all their licenses to the FDA before they'll approve that you are capable of running the study, you have the expertise, you have the team involved, and you have the whole environment set up. So that's the application to for the IND number. Now, the other thing I want to mention is intellectual property. And for those of us doing research, we don't often think about this because we're thinking about that deadline to get your abstract submitted. But before you submit your abstract, you need to talk to your intellectual property office, because as soon as you do your poster or your presentation, you have publicly disclosed your invention and you no longer can patent it. So it is very important, even for a poster or an abstract, that whoever, and I know they have intellectual property because I have some here with the NIH, that you talk to them beforehand and say, I'm submitting this abstract, is it okay or not? Or do you want to submit a provisional? Then if they say, oh, well, we're not sure, but we'll submit a provisional application for a patent, it only costs them like $500. They'll submit a provisional application. And with the provisional patent, that gives you a whole year to decide, is it worth submitting a full patent? But you're protected, and you can go out and you can present your research at ASCO or AACR or whatever. But that's important. So, and the other reason why it's important is if you don't have patent protection on it, when it gets to the point that your patent issues and you want to license it, or if you don't have a patent, a pharmaceutical company is not going to license your patent unless it has patent. They won't license it unless you have patent protection. They're not interested in developing your drug for the clinic unless they can make some royalties off of it. So, you know, as scientists, we don't usually think about this part, but it's important because if you're going to eventually develop a drug and take it to clinical trials, a pharmaceutical company is going to want to know, if I'm going to get into this, if I'm going to put money into your research project, am I going to get something back? So they want you to have a patent. So the other things that you have to do when you do a clinical trial, well, you got to write a protocol. You have to write a consent form. If it's a new drug, you have to also write the investigator brochure, which tells all about the studies that were done preclinical in animals, any studies that have been done in humans for other indications, and all about the drug and its pharmacokinetics. So even if it's repurposing an old drug, if there's not an investigational brochure available, you've got to do it. And then you get IRB approval. The hard part also is getting funding to do your studies. We all have, you know, great ideas and great projects, and we want to do this study, but then we apply and you don't get the funding to do it. You know, I had a project this past year, and a week before it was ready to start, AstraZeneca decided, I should watch what I'm saying, they decided that they're not interested anymore in pancreatic cancer, and they pulled out. So, I mean, you have to, you know, yeah. So anyways, it's getting the funding to do your project, you know, and whether it be any pharmaceutical company, I mean, but lots of times we're applying to the NIH or the DOD or somewhere to get funding to do your study. So the other important responsibilities is that the principal investigator has to do city training. You guys all know what city training is? If you're going to conduct a clinical trial, you have to do ethics and IRB training online for your institution, and you have to pass all their little modules in the city training to show that you understand the ethics about doing human research and what is HIPAA and all those things. So those are important, that you have to be city trained and approved, and then you hire a coordinator or a nurse that helps run your study. And then before you enroll the first patient and have them sign the consent form, you've got IRB approval, you got FDA approval, you got the drug, you have to register your trial on the clinicaltrials.gov website. If it is not registered and activated on the clinicaltrials.gov website and you have somebody sign a consent form and start your study, you're not allowed to publish your results. So those are important because you want to publish what you do. So you have to do everything in a systematic way. So that's a little bit. So what do you do as far as, we're busy working in mice, and my mice weigh, you know, 20 to 25 grams. But then how am I going to calculate, I'm going to take this compound and I'm going to give it to a human being. And how do I figure out if this is safe, what's going to be a toxic dose, and where am I going to start for figuring out the maximum tolerated dose? So I did a study with this drug called OGF many years ago, and it was the first in human study. And I started out, I figured out the dose, so I'll show you the formula how we do that, and then you kind of reduce it and you do your first dose. And I gave the first patient their 25 milligram per kilogram dose intravenously and she had metastatic pancreatic cancer. And that weekend, she ended up in the emergency room with abdominal pain. So I had my first adverse event on my first patient. And I had to make a decision because, you know, I could have stopped right there and said, oh, it's too high a dose. But we were doing the 3+3 dose escalation to figure out the right dose. And so I went ahead and dosed the next 2 patients at the same dose that she had received, and they did just fine. So, you know, I had to make a clinical decision. Was her side effects and her abdominal pain related to her cancer, or was it something that my drug did? So, you know, sometimes you have to make those decisions and decide, and the safety monitor helps you make those decisions. Are you going to redose patients? So we did, and we ended up finding out our maximum tolerated dose was actually 10 times higher than the dose that she had received. So if we had stopped at that dose or lowered, we never would have gotten the drug through all the different phases. So, so the way that you kind of calculate out the human dose, there's this table, and I hope you can read it in your handout there. But depending on which animal you used and how much the animal weighs, there's a different formula that you can use down here to kind of calculate out what would be the best dose to use in humans starting off. So there is a kind of formula that you can use to do this. So I was going to talk to you just a little bit about some of the research that I'm doing and how we're taking this from bench to bedside. So the work that I do is with cholecystokinin or CCK receptors, and they're G protein coupled receptors, which are the 7 transmembrane spanning receptors. And the main ligands that bind to these receptors are cholecystokinin and gastrin. So there are 3 main types of cholecystokinin receptors, the A, B, and the C receptor. The C receptor is one that our lab cloned, and it's only found in cancer. But they have different affinities for either CCK or gastrin. So we found that these receptors are overexpressed in cancer, but then we also interestingly found that they're also expressed on pancreatic stellate cells or fibroblasts that are in the pancreas. And they have both CCK-A and B receptors. So this is a mouse Pannin lesion, which I'll show you some of those. That's a precancerous lesion in the pancreas. And they develop CCK receptors as the cancer starts to develop. But surprisingly, we also found that the stellate cells had these receptors. And we said, so what are they doing on the fibroblasts? So we did an experiment using a CCK receptor antagonist. And there are specific antagonists that go to the A receptors and ones that block just the B receptor. And they have different affinities. But then there was this one called proglumide. And proglumide actually inhibits both of the receptors. And since I told you that they have both the A and the B receptor, we thought, well, maybe we'll try the proglumide. But I can tell you, honestly, one of the reasons why we use the proglumide is that it's water soluble. And we were going to be doing a study over 4 months period of time. And so I could put it in the mouse's drinking water. These other drugs, you have to dissolve in DMSO or some other, solvent, and you have to inject them IP a couple times a day or a few times a week. And I didn't want my technicians to have to come in on the weekend. So part of the reason that we use this, honestly, is it's water bioavailable, and it hits both receptors, and we didn't have to come in on the weekend to inject. So, it was a win-win situation. So what we did is we took this proglumide, we put it in their drinking water, but we waited until these mice were about 3 or 4 months old. Now, I don't know if any of you have ever worked with, these are KRAS transgenic mice, and over time they develop pancreatic cancer. And they do that by developing what these are called pannin lesions. And these pannin lesions turn into pancreatic cancer over time. And it's because they have a mutant KRAS. And so we waited until we knew that these mice had pannin lesions, which was 3 or 4 months of age. And then at that time, we randomized the mice and we put some of them on just regular drinking water. And some of them we put on drinking water that had proglomide in it. And we found that the pannins actually regressed. And this is a control mouse. And the blue staining here is a Mason's trichrome stain staining the fibrosis. So we found that not only did the CCK antagonist proglomide decrease the number of pannins by acting on the CCK receptors on the pannins, but it also decreased the fibrosis by acting on the stellate cells in the pancreas. And so we had less fibrosis, and these were mice that were up to 8 months old. So 4 months of treatment, less fibrosis and fewer pannin lesions. So we said, huh. So that was in the pancreas. And then I thought, well, what about if we grow these pancreatic cancers subcutaneously? You know, are these CCK receptors also on fibroblasts that are not in the pancreas, but somewhere else, you know, under the skin? So we did that. And lo and behold, we found, again, all the blue staining is the fibrosis. And the tumors of the mice that got the proglomide didn't have the fibrosis. And we saw that it was significantly decreased. So this drug has anti-fibrosis properties. So in pancreatic cancer and in other types of cancers, one of the problems, especially in pancreatic cancer, in treating it is that it doesn't respond to chemotherapy or immune therapy. And it's thought the reason why, one of the reasons that pancreatic cancer doesn't respond is because it has this dense fibrosis around the cancer, or this desmoplastic reaction. So we took our mouse model and we said, what if we can get rid of the fibrosis, and then let's see if they respond to immune checkpoint antibodies. So mice typically, or pancreatic cancer does not respond to immune checkpoint antibodies, but we went ahead and we treated the mice with our proglomide. And we found that when we did that, we actually increased the number of CD8 cells that were able to get into the tumors, and we shrunk the tumors. So down here is the combination treatment. But you can see that PD-1 by itself has absolutely no effect on the fibrosis. Only when they got proglomide, the CCK antagonist, along with it. So we thought, this is great. We might be able to improve our therapy for cancers if we can improve the penetration of the chemotherapy into the cancer. So I took on another idea. So we figure we have this drug that might get rid of fibrosis. Well, one of the biggest fibrosis problems in the world is cirrhosis, or liver disease. And non-alcoholic fatty liver disease, or NAFLD, and NASH, which is non-alcoholic steatohepatitis, is actually becoming the #1 cause of liver cancer in the United States and actually worldwide. It's becoming an epidemic because of the obesity epidemic. And now that we're able to cure hepatitis C and control hepatitis B and control HIV, the people who are now getting liver cancer are the people who all have NASH. And there is no treatment for that unless they lose weight. So this is the new epidemic of this millennial. And it looks exactly like on the histology like they're drinking alcohol, but these are people who are not drinking alcohol. And on the biopsies, you have the classic finding for histology is that they have inflammation, fibrosis, they get cirrhosis, and they have fat, which is steatohepatitis. So this is a normal-looking liver. We don't like seeing it out of the person unless we're doing a transplant. And that's the normal histology. This is a fatty liver, and that's why it looks kind of yellow, because it has fat in it. And you can see these holes in the histology. Those are fat droplets. And then the difference between just a fatty liver and steatohepatitis is that there's also inflammation, and there's what we call balloon degeneration. If you can see, there's like a hole with the nucleus is kind of fallen over to the side. That's apoptosis. The liver cells, the hepatocytes, are dying because of the NASH. And over time, this can turn into cirrhosis. And cirrhosis, as you can see, has that same pattern that we looked at with my pancreas mice. This is a Mason's trichrome stain. There's a lot of fibrosis, and there's these nodules in the liver with the fat. So NASH is becoming the #1 reason that we're getting liver cancer today, and also it's replacing hepatitis C for the #1 indication for liver transplant. So what are the causes of NASH? Well, if people are not drinking, then it's ASH is drinking. That's alcoholic steatohepatitis. So NASH is non-alcoholic steatohepatitis. And the 3 leading causes are obesity, which is 1/3 of our country, hyperlipidemia with high cholesterol and triglycerides, or poorly controlled diabetes. And this is our metabolic syndrome. How do you treat NASH? Well, you tell people to lose weight. I can tell you, they come into my clinic and I say, go lose weight. And they come back and they weigh 5 pounds more. They say, that diet just didn't work. So anyways, there are some drugs that have been tried. Metformin, if people have diabetes, we prefer the metformin because that's safer on the liver and the pancreas than some of the other drugs. Lipid-lowering drugs are actually good. We get a lot of consults that primary doctors send patients to me and they say, this person's taking a statin and their liver enzymes are high. Should I stop the statin? But actually, they probably need more of the statin to get the fat out of their liver. There are some situations where statins can raise the liver enzymes, so you have to use that with some scrutiny. Antioxidants. There was a study that was conducted here by the NIH, and Dr. Jay Hoofnagel was in charge of that study, called the Pivens trial. And the Pivens trial combined the pioglitazone, there's pioglitazone, somewhere here, pioglitazone with vitamin E or by themselves, and they did that in patients with NASH. And what they found is they actually improved NASH, but they didn't reverse the fibrosis. So, and vitamin E was used in that study at 800 international units a day. Sometimes if I use it in my practice, I only use 400 because at the 800 there have been some reported instances of intracranial bleeding and strokes. So, because vitamin E does thin your blood. So that's one thing to be cautious of. But this study, they did not include any diabetics. And there's 2 reasons for that. One, they wanted to make it a pure study, but there's a concern about putting diabetics that might have retinopathy on something that thins their blood, that they might bleed into their eyes and go blind. But because it was never tested in diabetics, we do not use vitamin E in our diabetics. So those are the treatments, main treatments for the treating NASH. But the problem that we have here, so liver cancer and also pancreatic cancer is on the rise, and they're predicted to become, next to Dr. Moody's tumor up here, the 2nd and 3rd leading cause of cancer-related deaths. So we did all of our work in the pancreas, and we showed that the pancreatic stellate cells have these CCK receptors, and we can block their receptors and decrease fibrosis in the pancreas. So we came up with this idea, and we hypothesized that, since pancreatic stellate cells have CCK receptors, well, maybe the stellate cells that are in the liver also have CCK receptors, and if we block them, we might be able to decrease fibrosis in the liver. So no one has ever described CCK receptors in, the stellate cells in the liver. So we designed a preclinical trial with our mice, and we had a prevention study and a reversal study. And we put these mice on a high saturated fat diet that was choline deficient and a thionine supplemented, because that also induces cancer, okay? And then we had the mice on this diet, and they actually got proglomide or regular water, and we had control mice on a regular diet without the high fat. And then this one, they started the proglomide at the same time as we started the diet. So this was a prevention study, and they were on this for, we checked some mice at 12 and some at 18 weeks. And then after we knew that they had fibrosis by 12 weeks, we actually took another cohort of 30 mice after they had known NASH and fibrosis, and we randomized them to say, okay, now you've got it, can we reverse it? Because I can tell you, people don't walk into my office over at Georgetown and say, I want to prevent my liver disease, what can I do? No, they come into me and they already have NASH and they already have fibrosis, and they want to know, what can I do to make this better before I get a transplant? So we want to know, can we reverse it? So we had half the mice on proglomide to try to reverse it. And so what did we find? Well, we drew blood levels on all these mice, and I don't know if you guys are good at drawing blood on mice, but it's we checked and we showed that the liver enzymes actually went up in the mice that were on this high saturated diet. Control mice, there was no change. But we showed that we could prevent this increase in the liver enzymes, and also we could reverse it after it already happened. The other thing that we found is bilirubin went way up, and we also reversed that and prevented that. So it had biochemical changes. And one of the criteria for NASH is inflammation, and inflammation means their liver enzymes are up. So we had a histologist look at all the livers from these mice, and they scored them in a blinded fashion and found that the proglomide decreased all of the three characteristics of NASH, the inflammation, fibrosis, and the steatosis. And this was a liver from one of our mice that was on the diet, and it did show the balloon degeneration, the fibrosis, and the fat, whereas the mouse that was on the proglomide, it was nearly normal. So then the most surprising thing at the end of our study at week 18, we found that 35% of the mice that were on the high-fat CDE diet got liver cancer, and none of the mice on the proglomide, whether they got it from the beginning or whether they were those mice that we reversed it after they already had NASH, and we only gave them 6 weeks of the proglomide after they had established NASH, but at week 18, none of them developed cancer. So we were able to prevent cancer too. So we thought, this is good. So we're going to try a clinical trial with this. So this proglomide drug that I talked to you about is an old drug. It was developed about 30 or 40 years ago, I think, to treat peptic ulcer disease, because CCK receptors are responsible for gastric acid secretion in the stomach. But then about 30 years ago, they came out with proton pump inhibitors, you know, the omeprazole and Protonix and all those drugs. And those were much more effective at decreasing acid secretion. And so the companies that were developing proglomide gave up on it. And they said, we're not going to do it anymore. But proglomide had already made it through clinical trials. They knew that it was safe. They knew what dose they could use. And so I have all that information where to start my trial. So, of course, before we submitted our abstract to be presented last year at this past April at AACR, we had to submit intellectual property, of course, at Georgetown. I got IRB approval. I have an FDA IND number. And actually, we just secured funding from the NCI to carry out this phase 1 trial, and our paper is recently published. So we're taking this drug, and we're going to test it to see what's the best dose to use in patients. So as far as the phase 1 trial, what we learned about that is we're not looking yet at efficacy. I want to figure out what's going to be the best dose for me to use in my phase 2 trial. So we're taking 3 different cohorts of patients and testing 3 different doses I'm going a little bit below the dose that was initially tested in patients for peptic ulcer disease because these patients have sick livers. And I don't know if the drug might be metabolized differently in somebody with a sick liver. And then we'll escalate the dose and see how they do. We're just looking for safety, and we're not looking at efficacy. If this works, then we go into the phase 2 trial. And then we'll compare it to something. And the FDA requires today that you have to show that your NASH drug reverses fibrosis. There are several companies, including, I can mention Gilead's drug, made it into phase 3 trials, and it just failed because it did not meet the endpoint for decreasing fibrosis. Because they know now it's the scar tissue or the fibrosis that increases the risk for liver cancer. It's not the fat. People could have fat in their liver, and that doesn't increase the risk. But if you have the fibrosis and the inflammation, that's what increases the risk for cancer. So that's one of the criteria for the FDA of getting your drug approved. So, and then if we find that it actually works in this condition and we can reverse fibrosis, then I'm going to take on cirrhosis or take on something else. I can put our liver transplant guys out of business. But anyways, but if it does have anti-fibrotic properties, because we showed that it works subcutaneously, so it's not just in the liver, it's not just in the pancreas. It has this fibrolytic property elsewhere in the body. So the obstacles of doing translational research today, money, of course. I don't know if it's because there's a lack of funds, misuse of funds, disparity of funds. It's hard to get the funds to do the trials, but it's not impossible. So don't give up. Apply for those pilot grants. Do all those things. I'm a clinician, so I don't know how many of you out there are clinicians, but there's this thing called protected research time. You know, in the clinical realm, it's all about how many RVUs you make. They want to know how many colonoscopies I can do a day because that makes more money than me bringing in an NIH grant and doing a clinical trial. Unfortunately, that's the reality of being out there at the universities and being a clinician. They want to know how much money you can make and how many patients you can see in an hour. No matter where you are, it's the same way at Harvard as it is at University of Pennsylvania, as it is at University of Maryland. It's all about the RVUs. So, but if you do get a grant, you put salary support on your grant, and then they are required by law to give you protected time for that amount of time that you put on your grant. So that's important. I say that there's this chasm between industry and academia. Of course, industry likes to develop their own drugs, and you know, but it's better if they would partner with universities and NIH, because that's where a lot of the great science is being done, not just at pharmaceutical companies. So, you know, I think they all need to come together, but I can tell you when I was working at NIH, NIH didn't want me talking to pharmaceutical companies because there was this division there, and it really shouldn't be that way. They all need to work together. There's always problems accruing patients into studies, and most importantly, there's no more one-man bands. The PhDs got to work with the MDs, and it's all about team science. There's absolutely no way I could do this without all my collaborators and people that have helped, you know, along the way. And there's no problem with, you know, you come up with a great idea in the lab to work with a clinician to get the study done. So these are just a couple of my patients that were in some of my pancreatic cancer studies, and with their permission, just to show you that what is the outcome. The bottom line is that this research really does help patients. You know, these patients actually came to me after they had failed chemotherapy and there was nothing else left to offer them. They enrolled in my study and they actually lived an additional 2 years, which was good for pancreatic cancer once you fail therapy. So, and this is just some of my research team over at Georgetown. And thank you. Any questions? In the back?
Thank you, Dr. Smith. It's a very quick talk. I do have one technical question for you. So I noticed that you put the drug into the water bottle to keep the line. Then how can you figure out the dose that actually gets taken?
So we know how much we're putting in. So the question was how much, I'm repeating it in case anybody's listening. Yeah. How much dose how do we know how much we're putting in the water? Well, we figured out how much we're putting in the water bottle, and we actually measure how much the mice drink. And we have typically 5 mice per cage, and you divide it up based upon how much is out of, we measure the water that's in there, and we know the concentration of the drug. And we had to do that on a weekly basis to see how much they're consuming and what is the right dose to put in the water. That's kind of how we did it. So, yes. Correct. That's true. And if you want to be very specific about it, you could probably do single cages and have a mouse like that. But we knew that the mice were all drinking it because we ended up getting histology. And the histology showed us that they had an effect related to our drug. So, we were looking for a response. It's expensive to have one mouse in a cage, and they like to commune together. So, you have to justify why you're going to separate the mice to your IACUC. But anyways, thank you. Yeah. Yes.
I think when you try to do some preclinical translational study, you publish your paper, your result. I want to know in which step you can publish your paper. I think because just after you get our IRB approval, or just in which step you can publish your paper.
You mean for the clinical study or the pre?
Yeah, if you publish paper first, and then you can do that.
So for the animal studies, you don't want to publish anything until you get intellectual property, or even go out and present it at a meeting, publicly disclose it. And then as far as publishing your results, you just have to make sure that before you start your trial, that your trial is registered on the clinicaltrials.gov website. And it has to be registered before you start the trial and have the first person sign the consent form. If they don't, if you have them sign and then you register the trial, then you can't publish your results. Is that what you're asking?
Yeah, only when you get the register number, then you can publish.
Yes, so you have to register. Every clinical trial in the world is registered on the clinicaltrials.gov website. And it has to be registered on there before you initiate your study. And I can tell you that I went to submit a paper to the New England Journal of Medicine, and they specifically asked me the date that I registered and the date the first person signed the consent form. And they check those things when you submit your papers. Yes.
Yeah, the news was just released that ranitidine, the anti-acid ranitidine, contains a carcinogenic that causes gastrointestinal tumors. So my question is, clinical trials were done, and then 60 years later, we discovered that this is not a safe drug. It has consequences. So I would like you to comment on the short term in which a clinical trial is conducted, the short sample of people that we use for our trial, and how do we manage these long-term consequences? How do you see the entire picture?
I did hear the news about Zantac having a carcinogen in it, but I think that was a contaminant from the place where it was being manufactured in India. But it is true that some of those drugs will raise gastrin levels because they block, acid. And whenever you block acid, gastrin goes up. And some of those medicines, the proton pump inhibitors, do increase the risk of carcinoids and neuroendocrine tumors in animals and gastric cancers. And that's why the FDA does have a time limit on how long people are allowed to take them. And you're only supposed to take those medicines. We have different timelines. The maximum is up to 12 weeks to heal esophagitis, usually 8 weeks for gastritis, usually 4 weeks for a duodenal ulcer. And the only people that it's approved to use longer than 12 weeks are people who have Barrett's esophagus or erosive esophagitis. The caveat of that is these drugs are over the counter now. And so people, once they start them, they take them. The reason why they put those limits on them when they approved them is because they do raise gastrin levels, and we know that gastrin can stimulate GI cancers. So
how do we improve the overall world? I mean, how can we make a better world where the clinical trials and the long-term epidemiological and survival studies?
And a lot of it does happen post-marketing, unfortunately, and that's where you see the FDA does pull some drugs back post-marketing if they find out that there is a complication, and they have done that. They have done that, and you know, but we do have to have regulatory bodies that, you know, monitor this sort of thing. Yeah.
Okay, last question. Just one more.
Okay. I don't know who.
I used to work before in non-alcoholic fatty liver disease back in Kentucky, and we used to get a lot of patients, I guess the number of patients who are not obese but have NASH, which later on they defined that as NASH, associated steatohepatitis. Do you get those patients, and would you enroll them in your clinical trial?
So you said that they were defined as NASH. Or,
Yes.
okay.
And then when they look at them, they're not obese.
Right.
So this is a highly
Mhm.
contaminated, like in highly contaminated areas. Kentucky is one of the states that is active. They have a lot of, I guess, pesticides plants.
Right.
And a lot of the people in the rural areas are exposed to high levels of toxicants.
Mm-hmm.
And they would have the same symptoms as NASH. They would come with fatty liver disease, but they're not obese.
So you don't have, you can have fatty liver disease and you can have NASH without obesity. So there's, you know, 4 main things that cause fatty liver disease: alcohol, which that means it's not NASH because it's ASH, poorly controlled diabetes, and hyperlipidemia, and then the high BMI. So there are other things and toxins that can also cause fatty liver disease. And you're right, there can be toxins. But in order to have the true definition of NASH, NASH is a histologic diagnosis. And when you do the diagnosis, you have to have evidence of liver cell death by apoptosis, inflammation, and the fat in the liver with fibrosis. That's NASH. Now, in our study, when we do a phase 2 trial, the FDA requires for all phase 2 trials that patients get a biopsy to confirm histologically. For our study, we're not doing biopsies initially, but they have to have elevated liver enzymes, abnormal ultrasound showing that they have fat in the liver, a FibroScan test done that shows that they have fibrosis in the liver, and they have to have one of those 3 conditions: a BMI that's elevated, high lipids, or poorly controlled diabetes. So odds are they probably have NASH. But for a phase 2 trial, when you're looking for efficacy, you have to use the histologic diagnosis. Okay. Thank you very much for your time.